Welcome back. We have just discussed the different hierarchical structures of a protein or a polypeptide. Okay, primary, secondary, tertiary, and quaternary. Remember, all proteins will not have quaternary structure. It is only for those proteins which are present in multimeric forms. Okay. Now, the basic question is, because whenever we have talked about these local conformations, the local geometries, alpha helix, then beta sheet, beta turn, all these things. But the question is, if you want to make these molecules, or think of this, that if you want to make different geometries, what you did is rotation around bonds, because unless you rotate, you are not able to rotate the bonds, you cannot get different shapes. So, there must be a lot of rotational flexibility in a polypeptide chain. So, now this in this session we will first try to show those that which bonds in the polypeptide are free to rotate and which bonds are rigid in the polypeptide chain. Because if it is, some must be rigid, because you are not getting infinite number of conformations, you are getting only a finite, ultimately the protein takes a particular unique shape. Okay. So, that means there are many bonds which could be rigid. At the same time, there are different bonds which are flexible, which allow it to ultimately come to the energy minimized ultimate 3-dimensional conformation. Okay. So, we should try to identify what are these bonds. Now, a protein is basically is having a peptide bond. No, first we will say this is carbonyl. Then you have this NH and then a side chain again here, and there is stereochemistry here, suppose R1, and there is a stereochemistry, and the stereochemistry we know that is L. Okay. Then again the whole thing repeats. So, there will be a CO here, then NH, like this. Remember, this is the bond, which is rigid, the CONH, because in this NH is anti to the carbonyl. This takes a transient conformation, and that is quite rigid because of the double bond character between the carbon and the nitrogen. So, that part is rigid. So, if that part is rigid, that means you have again another rigid part. So, this is continuously coming one after another, but in between there is this alpha carbon. In between there is this alpha carbon, and this alpha carbon is connected by single bonds, and there is no question of any resonance or any double bond character here. Okay. So, these are the bonds which can rotate. So, this can rotate, but keeping the whole thing together, you cannot disturb the dihedral angle between this carbonyl and the NH, that is still there, that should be in plane, and that dihedral angle should be 180. That anti conformation you should maintain here, but what you can do, you can take the whole thing and then start rotating around this C alpha and the carbonyl. At the same time, the same thing is applicable, that this C alpha and this nitrogen, this is a single bond. So, you can now take this whole plane containing the CO and NH, and you can rotate this in one direction, clockwise or anti-clockwise. So, these are the bonds which can now rotate, and that is the genesis of different conformations of polypeptides. Okay. Now, there was one famous Indian biochemist, Sir G.N. Ramachandran. So, he tried to see that whether all types of rotations or any angle of rotation is allowed or not. See, because when you do these rotations, what will happen, that the interrelation between these carbonyls will change, and also the interrelations between these R1 and R2, that will also change. Because if you start rotating, I will show you, when you rotate all these, so all conformations may not be permissible. Ultimately, he came to a conclusion that all types of rotations are not permissible. Okay. Now, before doing that, he defined two angles, these two angles. One is the, see, you have a framework which is planar, that means this carbonyl and this carbon nitrogen, this carbon carbon bond, this is the backbone, sorry, this is the backbone of the protein. We are just neglecting the substituents, we are talking about the backbone. Okay. So, this backbone, if you talk about the rotation about this carbon nitrogen bond, so what you are doing is that the angle between this bond and this bond, earlier it is now anti to each other, you are now changing that angle. Now, that is what is called the dihedral angle. Dihedral angle is basically between two planes. Now, usually books will write that the carbon nitrogen, that dihedral angle is called the phi angle, and the carbonyl carbon and the alpha carbon, that angle is called psi angle. But remember, these are dihedral angles. Dihedral angles are exactly basically between two planes. Two planes intersect, and that creates a dihedral angle. So, what is the dihedral angle? Because two atoms cannot make a dihedral angle. Although we say that this carbon nitrogen makes a phi angle and this carbon carbonyl makes a psi angle, but what is this phi and psi? Actually, what happens, that when we talk about phi angle, we take this carbon, maybe I can take another color, we take this carbon, this nitrogen and this carbon, that is one plane, and we take this one and that one, that is the other plane, and the common bond between this is this one, this carbon nitrogen. So, that is the dihedral angle, that is what is called the phi angle. Okay. Similarly, when we talk about the psi angle, it is basically the angle between the psi angle means this one. So, the common bond is this one, carbon and carbonyl carbon and the alpha carbon. So, the dihedral angle will be between the plane containing these and the plane containing these. Okay. That is the phi angle, again I repeat, and this is the that is the psi and this is the phi angle. Just saying that it is the angle between the carbon and the nitrogen is not enough, because dihedral angle is plane. Again I repeat, what are these planes? For the phi angle, the planes are basically this carbonyl carbon, the nitrogen and the alpha carbon, and the nitrogen, the alpha carbon and the carbonyl carbon on this side. And for the psi angle, it is the nitrogen, then the alpha carbon and the carbonyl carbon, and here on the right side the nitrogen, the carbonyl carbon and this carbon. Remember, when I am checking the two planes, we have a common bond in between. That is the way to do it. Now, let us show you the model, that what happens here. Here it is in the slide. If you go to the slide, see, I do not know whether how clear it is. It says that this is the psi angle and this is the phi angle, but what I am saying, that actually we should say it is the angle between the plane containing this carbon, this nitrogen, this carbon and the plane containing this carbonyl carbon, this carbon and this nitrogen. Okay. That is the angle which is not reflected here. The common between these two planes is actually this carbon nitrogen, and the common between the when you consider the psi dihedral angle, the common line is this one, this carbonyl carbon and the carbon. I can show you very simply that when we talked about the geometry of just N-butane, remember what was the dihedral angle between the methyls? We used to say the gauche, there is this dihedral angle in the gauche butane form is 60 degree, and sometimes casually we used to say the methyl dihedral angle is 60, but actually truly speaking we should say that the dihedral angle between these the plane containing this methyl, this carbon and the back carbon, and the other plane containing this methyl, the back carbon and the front carbon, that is what is the dihedral angle. Okay. So, that is why that needs to be very clear. Now, this question is whether all phi and psi angles are allowed or not. So, Ramachandran plotted this phi and psi. In one axis he kept the phi angle here, this is the phi, and on the other axis it is the psi angle. Okay. And you see that he actually bisected it into tetra. This is basically bisected two lines, horizontal lines. This is the 0 line, that is the other 0 line, that means here phi is 0 and here phi is 0, and this is, see, a rotation means a complete rotation, that will be 360 degree rotation. He considered that rotation from minus 180 to plus 180 for the psi angle, and this axis is minus 180 to plus 180 is the phi angle. Okay. So, from this side to that side phi angle is plus, from this side to that is minus, and from this downwards the psi angle is minus, and from the up the psi angle is plus. Now, he has shown these some regions which are red in color, some regions which are yellow in color. What does it say? And some are not colored at all, only white. Okay. What he found, he studied all these different structures, the beta sheet structure, the alpha helix, the turn structure, everything, and then he determined what are the phi psi angles in that. And what he found, that the red regions here, that means this is a point basically here in the red region represents a particular phi and psi combination. So, what he said, that this red region is the most allowed region combination of phi psi angles. That means, the peptide will assume the phi psi angle in such a way that falls in this region. Okay. That is the ideal case, the red ones. So, the red ones, what it says, the allowed regions, namely for the alpha, here it is A, it is alpha helix and beta sheet conformations. So, now this one is for beta sheet, that means for beta sheet the dihedral angle should be the phi angle should be here. No? It is say it should be at least we can say it cannot be 0, because the white region is sterically disallowed for all amino acids except glycine. Glycine does not have any steric effect. So, this is the beta sheet, that means beta sheet formation will be favored when there is a combination of something. If we take the top one, that means 180 and close to 180 is allowed for beta sheet formation. And for the alpha helix, this is the red region. The red region means, you see, that means alpha helix is allowed when the phi psi combination is close to what, means this is about, I can say if it is 0, this is about 40, 50, 40, the whole thing is 180, and on this side you see this is about 90, this is about 60, 70 and that is about 90, close to 90, because this is 0, this is 180. So, the middle point is 90, here the middle point is 90, that means 90 is not is not that is not being taken up. Close to that is the ideal region. Now, apart from that, you have a yellow region, which is this is the limit. I mean, sometimes you get values which are within the yellow region, but the ideal ones are this one, the red ones. These are the ideal regions. Sometimes you get a left-handed alpha helix. Sometimes you can get. Okay, that is the red region, but these are much rarer. This is what he says. This is the limit. Limit means when he draws a line, he said that means if you determine the structure of a protein and see that the phi psi angle falls here, that means your structure analysis is wrong. That cannot be the structure. So, whenever somebody gets a structure of a protein, so immediately he computes all the phi and psi angle, and he has to satisfy this Ramachandran plot. Otherwise, that structure will not be accepted as correct. Okay? Because if your phi psi angle falls within the white region, that means there is something wrong. So, this is very interesting, a landmark work from an Indian biochemist or biologist, J.N. Ramachandran, and for every biochemistry book you will see the Ramachandran plot. Okay. Now, let us see that why this 0-0 combination, which we will just explore what we can see from this some extreme cases. 0-0 combination is not allowed, or 180 combination is not allowed. Why are this? That means there must be some serious steric effect or electronic effect that is operating in these combinations. So, let us see. We now I have a small peptide here. Okay. Usually the N terminus starts from the left side. So, I take the NH here. No, I think do the it usually it starts from CONH. If you have the amino acid from the left, amine is free, then the CO, then NH. So, I will do that way. Okay. So, CO, then NH, maybe this way. Let me see, let me first fix the direction in which you will be comfortable. Yes, I think this is. So, this is your CO. One thing one should remember, that the camera is looking at me. So, what is left in my case is right in the actual image that you are seeing. So, that creates little bit of confusion. So, let me try to again correct. So, this is a carbonyl, that is C double bond O, C double bond O, you can see two bonds here, and then there is this NH. Okay. Then there is the alpha carbon with this substituent here, and this is the hydrogen, the green one is the hydrogen. Then you have again another carbonyl, and this is NH. So, you have CONH here and you have CONH here. So, that means there are two peptide bonds. So, that means we are considering if there are two peptide bonds, we consider a tripeptide. Okay. So, let us first what Ramachandran said, that you first make the extended conformation. Extended means like when we discuss the beta sheet structure, we drew the amino acid the peptide chain in an extended form. I think I can quickly go back to that. When we showed the beta sheet structure, you see we wrote the peptide in the extended, fully extended form, as if like an Illyar hydrocarbon. So, similarly, according to Ramachandran, that first you consider the peptide in a fully extended form. If you do that, then everything, this carbonyl, this is the NH, they are anti to each other, that you know why, and then you have this alpha carbon, single bond, you have this carbon, that is another single bond, this is the carbonyl and this is the NH, and then again the alpha carbon. Okay. Now, the question is which one is phi angle and which one is psi angle? The phi angle is between the carbon nitrogen containing bond. Okay. So, this is the carbon. Now, I rotate it this way. Okay. Now, the phi angle will be between the carbon and the nitrogen. The nitrogen means this one, okay, the NH, and the psi angle will be considered between this is the alpha carbon and the carbonyl. See, this is a single bond, and I can rotate without disturbing the planarity, coplanarity of the carbonyl and the NH. Also, they are transitoid geometry. So, I can start rotating this. So, I rotate. I am not actually doing this NH and CO, they still remain anti to each other. So, again now come back to this is the full complete rotation. So, this is the extended conformation. So, here the phi and the psi angles, they will be what, 180. If this is fully anti to each other, then they are 180, because if you look at this, the dihedral angle like the two methyls, when they are opposite to each other, they will be 180. That means, this bond and that bond, that means, I think if you look at this way, this bond and that bond, they are anti to each other, and that makes it a phi angle. Now, this is the psi angle, this is the carbonyl, and this is the phi angle. Okay, this is the phi. So, when you talk about phi, you see this carbon nitrogen bond, a carbon carbon bond, and this bond. Okay, and then you can see it. It is little complicated. I think the complication is even more because the image is just you are seeing the mirror image of what I am holding. That creates more problem. But again I repeat, you see it first of all in the fully extended form. If you do that, then it will be in the zigzag. And here the dihedral angle combination phi psi is 180. Okay. And that you see that in the beta sheet conformation, according to Ramachandran, that around 180 and 180 is allowed. Around 180 and 180 is allowed. Where is Ramachandran? Yes. You see, this is the region. That is the 180 region, and this is the 180 region. So, that is allowed. Okay. Now, if you start rotating, just follow me. I am rotating this, changing the phi angle. Okay. So, I rotate the phi angle, this is 90-degree rotation, and then ultimately it comes to 180-degree rotation. So, when I do 180-degree rotation, I see one thing, that this carbonyl and this carbonyl are very close to each other. So, what will happen? There will be a lot of electronic repulsion between the two. Oxygen has lone pair, this oxygen has lone pair. So, that is why this is 180 and 0, that is not allowed. 180 and 0. You see in this plot 180 and 0, you do not see any point here, that is not allowed. Okay. Again I come back to the original, that is the anti conformation we started. So, I rotated this by 180. Okay. I rotated it by 180. So, it goes there. And now, I rotate the other one. This is the psi angle. By psi angle, I rotate without changing the, remember, without changing the NHCO. I rotate it by 180. If I do that, see what happens. Now, there is even more problem. So, I have rotated from 180 I went to 0, and the other one also from 180 I went to 0. Because we started, because Ramachandran said that the extended form has 180-degree angle. So, when I rotate by 180, I go to 0, and this is also going to 0. Now, 0 combination is not allowed, and you see 0 combination is not allowed. So, that is, so he actually calculated all these, computed all these, and determined crystal structure analysis, and from that he came out with this plot. I think that is what it is. If there is any difficulty, again we can come back and show what is this Ramachandran plot. Okay. Now, determination of this 3D structure of proteins, there are different techniques to determine the 3D structure of proteins. One is the best way is X-ray crystallography, that if you can isolate a crystal structure of a protein, then that is the ultimate. Sorry, where is it? Methods of determining complete 3D structure. Usually there are three types. One is X-ray crystallography, that is the most reliable one. Then there is NMR spectroscopy, very high field NMR. Through NMR spectroscopy you can determine the 3D structure of a protein. However, there are some disadvantages of NMR spectroscopy in that sense that you cannot handle very large molecules. However, at the same time there are advantages that if you cannot crystallize a protein, then you cannot get the crystal structure by X-ray. So, all these advantages and disadvantages, X-ray needs crystals, but and the structure is a static one, is the average structure in the crystal, but NMR is in solution. So, what you see is exactly the dynamics of the system. So, but you need highly concentrated solution to get the signals. It is actually very complicated ones. You should have isotopically labeled the carbons, isotopically labeled the nitrogens. There are techniques to do that, but by NMR you can do the structure analysis, but not very large proteins. There is a limit maximum about 60 kilodalton, up to 60 kilodalton. At present you can do the structural illustration. Electron microscopy, that started long back, but recently there is a technique which has won the Nobel Prize 3 or 4 years back, that is called cryo-electron microscopy, and through that many of the crystal structures are being solved these days. Okay. Now, let us come to, so we have done the structure analysis, we have completed the structure analysis. Now, we come to purification of proteins. Purification of proteins, first of all, for any purification method, you have to see what type of property these molecules have, so that you can utilize them. Okay. So, let us see what are the parameters that a protein possesses. One is the molecular weight. Some proteins are small, some proteins are big. When we talk about purification means, we have to separate the proteins. Okay. So, purification can be based on molecular weight if the proteins differ in size in molecular weights, or there could be charge difference, because we know that proteins have different side chains are charged. Okay. So, there may be difference in charges in different proteins. There can be difference in solubility in buffer or in water, or there could be some affinity for a particular agent, and we can utilize that affinity for that agent to separate protein molecules. Like if you are trying to catch a fish, you can particular fish in a pond. So, what you can do, if you want to catch a particular type of fish, then you should use a particular fish hook which the fish has affinity. Similarly, if you have affinity for a protein, so that you can take up the protein out. So, these are the 4 parameters that one can use. Okay. Now, the processes that are based on molecular weight, that means size of the protein. One is called the gel filtration, and the other is called the SDS page, that is sodium dodecyl sulfate and polyacrylamide gel electrophoresis. And by these two actually separate the proteins, a mixture of proteins as per their size, and the size is nothing but the molecular weight. Of course, there is an important point here, that when I talk about size, some proteins may be really globular in shape, and some proteins may be little bit stretched in a linear direction. So, then size may be different. A high molecular weight molecule may have lower size than a low molecular weight molecule. Okay. We will come back to that. Then the process is based on charge is ion exchange chromatography, native gel electrophoresis, and isoelectric focusing. So, we will consider all these 5. Let us see how many we can do today, then the next, otherwise we will do it in the next session. The first one is protein purification of proteins by gel filtration. Gel filtration is also called size exclusion chromatography. what it does, that you fill a column with some matrix which are like a molecular sieve. Sieve means which can filter out something, okay, a sieve, and like they look like small beads also, very tiny beads. Now, what you do, that you add the protein solution of different or any other macromolecule also you can do, polymers, even chemical polymers can be separated by this process. Proteins you add, and different, and depending on the type of bead that you are using, you can have a cutoff. That first of all you have series of beads in a column, and then you add the protein solution, and what will happen now, there will be a separation between the large proteins and the small proteins. Why is that? I think that will be clear in this slide. See, these are the beads that you are using to separate the proteins. Now, you have a mixture of proteins. You can see some green balls here, that is representing a green is bigger one, that is a bigger protein, this is a smaller protein. There may be others also. Okay. Now, as you have, these are all packed in a column, in a glass column. Now, as you add the protein solution, the next what will happen after few minutes, you will see that they have started dropping, because this is by gravity, they will slowly come down. You are adding a solvent and maybe water, and then this is coming down. Okay. Now, what will happen, the smaller ones because of their small size, there are many holes here, as you see, there are many pockets where the small ones will fit, but the larger ones cannot fit into those pockets. So, basically what will happen, the smaller ones will fill the pocket, and then as the solvent goes down, they will slowly come down. So, their velocity of coming down will be much less than the larger ones, because larger ones cannot fit into the small holes, and then as time goes on, you will see now the larger ones have come ahead of the medium ones, and the medium ones are ahead of the smaller ones, or the smallest ones. Okay. So, then ultimately what will happen? Yeah, you see the larger ones are almost now coming down coming away from the column. Okay. So, this is the principle of what is called gel filtration chromatography, and you have different sizes of these beads, and basically if you want to, they say a cutoff, means they will say that a 10 kD cutoff gel, that means molecules which are size less than 10 kD kilodalton, they will be attached more to the bead, and the others will flow through very quickly. Okay. Or you can get 40 kD cutoff, that means your protein of interest has a molecular weight of more than 40 kD. Okay. Then you use the 40 kD cutoff. So, this is a primary purification step that every protein chemist does, that gel filtration, that is first you determine the molecular weight, and then once you determine the molecular weight, you can determine the assess which type of this type of bead I should see. They are made up of carbohydrates called saccharose gels you can use. There are different cutoff levels are available, and then do this chromatography, size exclusion chromatography. Okay. Because the basis of separation is actually size. Okay. Then this SDS page. SDS page is again I said SDS is sodium dodecyl sulfate. It is a long fatty chain with a sulfonic acid, and page is polyacrylamide. It is a acrylamide that is polymerized along with a cross linker, and that makes a gel, like it is said, polyacrylamide gel. Polyacrylamide works like agarose is another gel. There are different types of gels available. Anyway, when you have this gel, in this case you do electrophoresis. Remember I said earlier electrophoresis is basically separation by applying a voltage difference, and when some compounds are placed in a matrix. So, according to the charge they will move, charge as well as their mass, they will move. So, this is the apparatus that is this polyacrylamide gel apparatus. What it does, you see that this is what is the gel, and there are these are called grooves here, where you can add your protein solution, and on this side there is something, a tiny test tube like thing. These are made up of actually plastic, they are called Eppendorf tubes, and this is your micro pipette, and there is a power supply. So, which keeps this is the anode and that is the cathode. So, what you do here, you first, if this is your protein solution, suppose lot of proteins are there, what you do, you add your SDS, that sodium dodecyl sulfate, you add it and then heat this to a certain degrees, not very much, maybe 60 to 70 degrees you heat. And what happens as you heat, the proteins were earlier in a globular, in kind of folded state, okay, in a folded stage like this. So, when you add this into SDS, these are SDS molecules, they are negatively charged, sodium dodecyl sulfate, that means it is a sodium salt of a big anion, okay, hydrophobic chain followed by a head group carboxylate. And these anions, now what will happen, as they are being mixed and little bit warmed, so they will, this protein will unfold totally, and it will be surrounded by all these SDS, its surface is surrounded by the SDS. So, this protein will also do that, that will also become linear and that will be surrounded by SDS page. So, now what will happen, the large proteins and the small proteins, there will be a difference in their length definitely, but the charge density will remain the same. The charge density will remain the same, okay, because every point there is a sodium dodecyl sulfate. So, if suppose this has got 10 amino acids, so it has got 10 sodium dodecyl sulfate on one side, that means per amino acid you have 1. If this has got 5 amino acids, it will also have 5 sodium dodecyl sulfate surrounding it. So, ultimately the total charge density on the surface will be same for all the proteins. Okay. So, now you have maintain similar charges in the proteins, and now you add it to the groove. As I said, on this groove, these are called wells also. So, you add the protein, and now after adding the protein you apply the voltage. As you apply the voltage, now what will happen, the charge density being same, now only the molecular weight only will be the deciding factor. This is a small molecular weight protein, that is a large, medium, that is large. So, the large ones will come most slowly, then the middle one, and the first one, the one which is moving ahead of the others. Remember, the current actually is, this is your anode, the positive end, because everything is negatively charged, surrounded by SDS. Okay. So, that is why this is called SDS page, but this is also called the polyacrylamide gel electrophoresis under denaturing conditions, because what you have done is you have denatured the protein, you have destroyed the 3D inherent 3D structure of the protein. But what you have done, so after this electrophoresis is over, what you usually have another dye you add, which moves ahead of all the proteins. So, when the dye reaches at the front, you stop the current and then you take the gel and put it in a dye which is called Kumasi blue, and then you can you stain with Kumasi blue, keep it in overnight in acetic acid methanol, and then wherever there are proteins you will get blue bands. So, these are the proteins that you see. So, that means if you have different Eppendorf tubes, so your tube 1 has got a major protein here, another major protein here, another major protein here. Okay. So, depending on the intensity, and also what you do usually, you put a molecular weight marker. You can give a standard proteins on one lane you can run where there are where the molecular weights are known. So, from that you can tell what is the molecular weight of the protein that that this protein has, because you have molecular weight markers on one side, either this side or that side, so that you know the molecular weight of each protein. Okay. I think that is the SDS page purification. You also have very similar native, you can have native gel purification also. Native gel, you do not add SDS, you just apply voltage in the gel, where the protein maintains its 3D structure, in its conformationally active form. But native gel requires lot of voltages, because first of all SDS gives lot of negative charges, charge density is very high. For native gel, the intrinsic charge ultimately will decide which way it will move, how fast it will move. So, but native gel will not be dependent on the size of the protein, it depends only on the charge. So, native gel electrophoresis is also there. If you want to see separate in the native form, then you have to see the native, you have to do the native gel electrophoresis. Otherwise, generally for identification purpose, we do the SDS page. I think the principle is now clear. So, the SDS page depends on the size of the protein, and native gel electrophoresis depends on the charge, intrinsic charge of the protein at the pH that you are doing the gel electrophoresis. Okay. Thank you very much.